Thanks also for the discussion. I think now we deserve a break before we move forward to our last session. We're a little bit behind schedule, but I think we can still afford half an hour, 20 minutes break, okay. Because 20 minutes break, we might see that because we have a presentation from our colleague from the US, they might have online participants which really connect to the point. So 20 minutes, see you then.
Okay, I'm getting the signal from the back. It's good to go. So welcome back, everyone, including those online. My name's Graham Plasto. I'm from the University of Alberta, one of the international participants. And it's a great pleasure to invite James Koltes to give us a US perspective. And I just wanted to say very quickly, we saw a very nice timeline this morning, even beginning more than 10 years ago. We know we're 10 years of FAANG. We started the discussions in terms of where we are today at PAG, at that meeting that Krista identified. And in fact, I think, and of course I may be wrong, but Eurofang began with meetings in Brussels where a key part of persuading the Commission to invest in Eurofang took place with some very persuasive American colleagues. So with that background, James, let's hear about where you are in the US.
Thanks, Graham, for the introduction. I would be remiss if I didn't say that it's wonderful to be back in Belgium. It was about 13 years ago I was here. I was a Belgian American Education Foundation Fellow, so my tie for the fellows, and so it's very special to be back and wonderful to hear about all of the awesome research going on in Beauvais. So we'll see if I have the same issues with the clicker or not. There we go. So first and foremost, I need to say I'm here representing a much bigger group. And really, these are the folks that have led the way. I've been a part of this project for not quite 2 years. And these folks have really carried the load. So the original PI was Pablo Ross at UC Davis. Huizhen Zhou has taken over as the PI on this project. And then there are collaborators from across the United States who've had important roles in collecting samples, running assays, doing lots of different work, as well as some collaborators from Canada. So just briefly, I'm going to give you a big picture overview of things, talk a little bit about some of the results and where we're going, and then leave you with kind of the what's next, as I think there's a lot more work to be done with FAANG for where we're going to use it and extend upon it in the future. So the overall project, this is a quick outline, and I need to thank Huizhen again. He shared a number of slides. The first aim really focused on characterizing gene expression, second aim on epigenetic markings, third aim on integration, although also the fourth aim on integration. I'm going to talk primarily about aims 1 through 3. Really importantly, I need to, again, acknowledge a number of folks, and as Krista said before, there's often folks that unfortunately get left off on this from the labs, so I certainly appreciate everyone. This was a list shared with me, and so I want to acknowledge folks from Zoetis especially who did a lot of sequencing for this project and contributed that as a gift. That was very important to extend the capacity of what this grant could do. A lot of what you'll see today is from UC Davis, but very important contributions, samples, planning of experiments, planning of the grant, and et cetera from University of Idaho, Washington State University, Virginia Tech, Penn State, collaborators at USDA ARS in Clay Center, Nebraska, postdocs at Iowa State University, and collaborators at Texas A&M. There we go. So as an overview, this is a list of the 47 different tissues and cell types that were evaluated in the Bovine FAANG project. You can see it's a fairly diverse number of tissues. There were both adult and fetal tissues, so that fetal part a little bit different than what was used in Bovreg. And the majority of these animals were closely related to Dominet, the animal used in the genome build, so primarily a Hereford breed based. There's also some mammary gland tissue from Holsteins. And of the epigenetic data collected for epigenetic markings, there were 40 adult tissues with 8 fetal tissues, both from the Hereford line, again, either from dominant or closely related relatives for the animal used for the genome reference build in cattle, and 4 primary cell lines, muscle and adipose-based cells, and then 5 different stages of development from the Holstein mammary gland. I believe this was led by Monique at Texas A&M. So there were a number of different assays. I mentioned the gene expression data, but to kind of put that alongside the data used to annotate chromatin states, here's all of that information. So 47 cell tissue types in the expressed regions, so trying to get RNA-seq, primarily mRNA, small RNAs, and the rampage to get the promoter start sites, and then something else unique, the transcription termination sites, WTTS-seq from Washington State. And then for the chromatin states, kind of the typical histone markings to get at different chromatin regions, as well as ATAC-seq, and then whole genome bisulfite sequencing on a subset of animals from 40 different tissues and cells. I'll come back to this, but those don't quite overlap exactly. As we were generating the epigenetic data set to give you an idea of the number of samples, and again, these are representative across tissues, you can see on the table to the left, again, those histone markings used, those epigenetic markings, along with the number of samples from adult tissues, fetal tissues, cell lines, and the totals, so ranging from about 57 to 80 total samples for the histone markings, and then whole genome bisulfite sequencing on 95 samples, 114 samples with ATAC-seq, and the WTTS, those 3' end verification sequences on 76 animals. This has 2 replications per tissue, and again, this is both adult and fetal time points represented here. And then on the right, you can see this heat map, and what we're representing there is just the relationship across these different epigenetic markings across the samples. In terms of annotating those epigenetic regions and trying to get at epigenetic regulatory region annotations, there was a combination of data sets used. So from the Bovine FAANG project, again, this is getting at the overlapping data to build the overall regulatory set. 27 adult tissues from Hereford, 5 fetal tissues from Hereford, the 5 Holstein tissues from mammary gland, and the 4 cell lines. And then a public Australian data set from Holsteins that included 23 adult tissues and 4 fetal tissues. Here's the breakdown of those specific samples amongst those tissues, sample numbers among those tissues, as well as the number of cell lines. So between 56 and 60 additional samples contributed from that Holstein data set to come to a total of, whoops, it did it to me, 46 adult tissues, 6 fetal tissues, and 5 cell lines available for annotating those epigenetic elements. So moving forward, talking a little bit about the results, one of the main outcomes of the project so far has been annotation of the transcriptome. So this publication is out and accepted now. We hope it will be published soon at GigaScience, but you can find it online if you search at bioRxiv. Looking at the project, about 35,000 genes annotated, 160,000 transcripts, of which 79,000 represent protein coding genes, with an average of close to 5 transcripts per gene, so pretty close to the recommended level, hopefully expected level from the ENCODE project. This is in comparison to Ensemble and NCBI, and I apologize with the updated Ensemble build. This is now a little bit dated, so these comparisons may be a bit different, but at least at this snapshot, the annotations were perhaps a little bit closer from the current Bovine FAANG project in the US to the NCBI annotations. So digging into this a little deeper and looking at the types of annotations, about 160,000 total transcripts annotated, so not quite as many as from the Bovreg project, 79,000, not quite 80,000 protein coding transcripts, and a little over 80,000 non-coding transcripts. And there you can see the breakdowns for the various small RNAs and other types of RNAs annotated. There were about 118,000 transcripts, or 73% of the total, that were structurally validated by the various independent assays, and I'll show a graphic of that here in just a little bit. 69% of the transcripts were previously unannotated. 86% of those unannotated genes were derived from existing annotations, and 14% were completely novel. So some observations about the gene and transcript expression across tissues, specifically between the adult and fetal stages. Fetal testes showed the highest number of expressed genes, while fetal brain muscle tissue showed the highest number and percentage of non-coding genes. If I remember correctly, for the testes, this is similar to the Bovreg results. Fetal tissues had a significantly higher rate of alternative splicing events than adult tissues. Fetal tissues had a significantly higher proportion of unique non-coding transcripts compared to protein-coding transcripts in comparison to those adult tissues. And there were 106 non-coding genes identified in fetal tissues that very interestingly switched to become protein-coding genes in their matched adult tissues, so showing that interesting change, and there's more ongoing analysis in this area because that's quite an interesting phenomenon of what's going on with the regulatory regions. So in terms of looking at tissue-specific transcripts, about 9% of all genes, or a little over 3,000, were identified, as well as 15,000 transcripts that were expressed only in a single tissue. Majority of those tissue-specific genes, about 75% and 84% in terms of the transcripts, were unannotated. Testes and thymus had the highest number of tissue-specific genes and transcripts. As we expected, the expression level of tissue-specific genes were generally lower than their non-tissue-specific counterparts. So getting into looking at some of these data sets, these upset plots are kind of interesting for that. Hopefully I can characterize this in an explainable way. But in terms of validating the predicted transcripts using independent data sets from different technologies, I'm going to show in this plot essentially the number of transcripts that we identified that were overlapping with the previous annotations, be it Ensemble or NCBI. Those are represented by that lighter green and darker green box. So you can see that's if you go across the top of the bar graph with those colors, you can see those various dots. Those represent the specific classes of transcripts validated by those previous annotations, and then all of the other different colored boxes that are here represent support from different transcriptional assays or epigenetic assays from the Bovine FAANG project. So lots of different comparisons here, but essentially showing there were quite a few transcripts found additionally beyond those that were at least previously in the published annotations. If we look at now the validation of predicted genes using independent data, again with those two green bars, they've moved here, but it's the same colors representing previous annotations, and the others from our transcript and epigenetics assays. Again, you can see majority of the genes identified were already annotated, but only 12,000 of the 35,000 total. I shouldn't say the majority, but the largest segment of genes identified. So there was quite a bit of information added by adding those other assays from the US Bovine FAANG project. Moving forward to get at the impact of the epigenetic markings, we needed to be able to obviously build the chromatin states and dig deeper into what those chromatin states mean in terms of development and phenotype and whatnot. One challenge is that sometimes you have incomplete states, so imputation was done. This work is from UC Davis, so you can see on the left this plot where you've got these green bars. These are areas where the chromatin data was present, and then in red was where we needed to do imputation. So you can see there were a number of places where these chip chromatin marks needed to be imputed. Excuse me. And so the plot that I'm showing shows the correlation between a truth data set and a masked data set. So these are samples from a data set where essentially the true results were removed, and five-fold validation, cross-validation was done to impute those markings. So in some cases, very high imputations of up around 1, and on the lower end, maybe around 0.5. From those cross-validations, the group looked at imputation accuracy by area under the curve and some different methods. And in general, they were fairly good, ranging from around 0.99 to a little bit higher than 0.75 on the low end. So based on that data, it was used in Chrome HMM to do chromatin state modeling. Initially, a 10-state model was conducted, so kind of moving from left to right on this plot, you've got those typical annotations of chromatin state that you're used to, along with their abbreviations here, the emission pattern from the 4 different sets of histone markings used. Then as you move kind of to the middle where it says TSS transcription start site, so you can see the relative location of those markings from the TSS. The genome coverage is the COV, and then the annotation overlap with existing annotations from gene builds, along with expression on the very far right. And then these colorful bars furthest over to the, I believe that's, I guess, yeah, to your right as you view it, are the enrichment of non-coding mammalian conserved regions. This is looking at GERPs in the genome, so those regions that we think are highly conserved, regulatory regions. But there were more histone markings available, so the group went forward and did a 15-state model, and so kind of moving from left to right, the left side shows how those given number of states were identified as what was the level to use. Again, these were from 2 replicates, so 15 states was chosen from the Chrome HMM model, and then those states were annotated, and so you can see those 5 extended states here from adding attack-seq data and CTCF data, as well as, again, the relative position of where these different markings line up in comparison to the TSS. So that's the data that's going to be presented here moving forward, and just wanted to make some comparisons of what that means to add those extra 5 states, at least from the US FAANG data set perspective. So you'll see on the left the core 10-state model with an example using one of our favorite genes, myostatin, and it is a very highly expressed gene in muscle. So I've tried to highlight in yellow, I'm not sure how well yellow it shows up, those tissues because it's kind of small text, but there's muscle tissues and muscle cell lines that are highlighted there. And you can see the TSS in the middle in red. When you go to 15 states, we can start to pick up some additional chromatin states, and we get a little bit of a cleaner resolution of some of those transcription start sites, not surprisingly, with more data. So looking at the open chromatin accessibility, these are indicative of regulatory regions, obviously, and so if we look at the chromatin states, again on the left from those different colored bars, we can see the TSS annotated regions align with these red bars for promoter states, the yellow aligning with the enhancer states, and the green with the bivalent and repressed states. So just as another example, looking at chromatin state in terms of tissue specificity, there's an example for an adipose-specific gene. This is, I believe, early B-cell factor 3, EBF3, and it's a adipose tissue-specific expressed gene. And so you can see by those plots at the very top, those are the adipose tissue graphs here showing those promoter regions, I believe. And it's showing that you've got adipose specificity for that particular gene compared to essentially no signal or very little signal from the other tissues. Just as another example showing tissue specificity of gene expression as contributed by tissue-specific regulators using, in this case, myosin-heavy gene 2. So that's a muscle-specific gene. If we look at the gene expression, you can see that it's very highly expressed in muscle and essentially almost absent in the other tissues. And when we look at the annotation from that 15 chromatin state model of the transcription start sites, and particularly for active promoters, it really flags those muscle regions. So it looks like these annotations are working like they're supposed to for some of these known examples. Of course, another interesting thing is comparing these different developmental states, looking at fetal and adult tissues. And so the next example I want to show is with some genes that are expressed in early muscle development, myogenic factor 5 and 6, MIF5 and 6. Not surprisingly, if we just compare fetal muscle and adult tissue on the left, these Venn diagrams, we can see there's some overlap, but they're largely quite different. And if we look at the figure on the right, looking at a specific marking of enhancers, you can see quite a difference between the upper portion of the plot, which has fetal tissue, the lower portion that has adult tissue for muscle, and then on the bottom looking at those specific annotated chromatin states, where you can see there's quite a difference between the top, again, the active promoter region annotations to the top, and then kind of the repressed regions to the bottom in the adult tissues. So we're also trying to work with DNA methylation data. A lot of that is data that's kind of hot off the press, at least in terms of analysis, looking at how that DNA methylation corresponds to chromatin states. And of course, we know that DNA methylation tends to repress gene expression, and so we can see that at the TSS here on the bottom left, there tends to be less DNA methylation. And if we look at the enrichment in some of these promoter element regions, it does indeed show lower methylation levels as we would expect. And just going on, digging into a specific gene example for a gene expressed in testes and as well as other tissues. I mentioned there's a lot of diversity in transcription in the testes, it appears, or at least in transcript abundance. What this plot is showing over to the left is that among the 208,000 differentially methylated regions identified, this is a particular gene region that was differentially methylated, so much lower at about 50% in testes and then quite high in the other tissues. And there was essentially a correlation of -1 between methylation rate and expression level, which is interesting because this gene is involved in spermatogenesis and reproduction, and being a gene that is lower in methylation status in testes, it makes good sense that it might have some kind of role in reproduction, tying it back to reproductive phenotypes. So in terms of what's ongoing, there's a considerable amount of work being done to evaluate those regulatory regions, where they are, how they are located in open chromatin and the different chromatin states, as well as to look at the impact of DNA methylation within these regions. And so this is a plot just looking at enrichment of some of these different regions within the different tissues of those transcription factor binding sites. With that, I mean, this is so far a lot of general characterization for the Bovine FAANG project. We've got that initial paper that's coming out, and there's 1 or 2 additional papers that we hope are coming soon, focusing on some of these ongoing research topics. So looking at allele-specific epigenetic regulation across tissues, characterization of regulatory elements, looking at tissue specificity, that switching event that occurs between the fetal and the adult tissues, or maybe in other cases across tissues, and evaluating super enhancers and enhancer modules, as well as trying to dig into the functional mechanisms of regulatory elements, integrating with GWAS data, and this is a collaboration between the UC Davis group and the cattle GTEx. There's also application of complex trait adaptive evolution, looking at conservation of regions, and trying to build open access data portals. There's a lot of data here. Some of that will become available very soon at the Ensemble portal, and there are additional plans both to make additional regulatory region available from these projects that are ongoing, as well as to integrate these data with the public resources at animalgenome.org, for instance, that's run at Iowa State. Maybe. It doesn't want me to go on. Well, whether it goes on or not, I'll tell you that the next slide is about needs. Oh, there we go. Or at least what I see as needs moving forward. And there have been a number of interesting discussions today, and so there's going to be some repeated topics here. This is an important subject, and I hope I'm doing okay on time. Graham, am I doing okay so far?
I've just been enthralled. So
okay. So some of these things I know are being done, and some are new. But one challenge is just keeping going. And so I know that from everybody I've talked to that's leading a FAANG project in the U.S. across all of the species, one challenge is that we've got a lot of data and we haven't gotten done everything that we wanted to, either that was planned or based on what was found to try to move science forward. So there's a lot of work that needs to be done and additional funding that's needed. One idea I would like to seed here is I think there's more opportunity for European-American collaboration, and there's a lot of work that needs to be done. So it's been previously mentioned, one of the needs is a regulatory element build for cattle that I think importantly, needs to help us catalog breed diversity. So that's been discussed quite a bit. There's very active pangenome and telomere-to-telomere sequencing projects going on in the United States that I've been fortunate to be involved in, led by Tim Smith, Brendan Murdoch, Steph McKay, and probably a bunch of other people. I apologize for omitting their names, but lots of folks, Ted Kalbflesh, who's submitted an initial paper on this that hopefully folks will see soon, going on, and I think this is where we need to dig deeper, because that's where selection has shaped what's happening in the phenotype, and that's something that will help us link genotype to phenotype, but it's going to take a lot of work, as Fergal mentioned. Additional work with single-cell data. So we've heard some folks talk about that already, and I think very, very importantly, how to integrate that back to our bulk data. The Bovreg group has done so much work with the bulk tissue data. The US group has as well. There's been some enriched cell data, but there's just so much work I think that's needed yet. More functional investigations. I was really happy to see the cell culture work that's being done in Bovreg. I think there's need for much, much more there, as well as CRISPR screens to dig deeper on functionality of genes and to try to verify some of these regulatory regions. More investigation of chromatin conformation, and I may be a bit short-sighted in this comment, but I think there's a lot of work to be done in terms of looking at the impact of genotype, looking at additional new molecular methods to phenotype regions that impact chromatin conformation, and to see how that impacts the overall makeup, be it at the molecular level or the observed phenotypic level. As well as outreach and promoting the use in the broader animal science disciplines. This is a focus of the US NRSP8 group, which is our kind of our cheerleading group to develop resources that are not always attractive to funding agencies, but to do things like build genomes, develop SNP chips, help develop FAANG resources. They've helped pay for some of the initial FAANG data, for instance, through that project. Now I think we need to go out and tell our animal science colleagues how great this is, help them to use it. Many of them already are. Lots of folks are using FAANG data. They just don't know to call it FAANG data. Some wonderful physiology was discussed today, and that interplay of understanding heat stress physiology or reproductive physiology in the context of FAANG data, I think is an incredible opportunity, and I think I see some opportunities between the US and European groups to work together on this, both to promote this and to raise awareness, and very obviously, hopefully, that will lead to increased funding as well for projects. And then I think after that large list, then I think we're in a spot to link genotype to phenotype to work in at breeding, and that's a very important goal and a very exciting goal. There's a lot of work to be done, I think, yet to get there. Some things it's wonderful to see that folks are working on, but much to do. Just some quick challenges I'm going to mention. There's a need for more information to link genotype to phenotype, thus see my list above. And a need for more bioinformatics students and postdocs, at least from the US perspective. Everybody that I talk to seems to be looking for students and having challenges finding them. The private market pays well, and it's hard to attract folks on a postdoc salary and getting students going early. Maybe we all need to focus more on training graduate students. We certainly can use more data savvy people. Not to detract at all from the biological data as well, because I think we're entering a time where the molecular biology to understand phenotypes is also very important, but we need people who can help with that data analysis, in some cases maybe do both types of analyses and interact well amongst the groups. So with that, I thank you and welcome questions.
Thank you very much indeed, James. So any questions? Do we have any online, Ricardo or Federico? Yes. And I'm hoping for a couple from the audience here as well. You mentioned in the intro that you have Australian collaborators, including one for cell line. So what was the cell line that was investigated in Australia?
So that's the data set from Amanda Chamberlain's group. I've got to admit, I don't remember all of the cell lines off the top of my head, but I believe there was muscle and kidney in there amongst others, but I'm afraid I don't remember those. If you'd give me a moment, I could probably reply to that question online in 5 minutes.
Right, and then there's another one, I think, where you sequencing, with the transcript sequencing, was that mRNA or total RNA?
Primarily mRNA.
Thank you. Krista? Yeah, where's...
Thank you for a great talk, James. I know why I invited you. One question regarding the data. It's also a tremendous amount of input. Will this all go to the FAANG Data Coordination Center with the same annotation we have? Because you mentioned that you will set up also some other portals. The portals would comprise then the secondary and tertiary analysis, or what would be the portals for?
Yeah, so that's a great question. So initially with the first publication that's at GigaScience, there's a large chunk of transcriptomic, rampage, WTDS, histone marking data that's currently at Ensemble, and the additional data is to become fully publicly available. There's a lot of data yet, and it's important that it is made public. It's just been slow to get that out. In terms of what will be available at the portals, most of it is going to be kind of secondary or tertiary analyses. The plan is to get the raw data out and to the public. But for instance, some work initially to do gene expression atlases to kind of in real time get some of this data available from data sets and combined data sets, looking at annotation of some of those histone markings, regulatory elements is something that will come out initially. And then there's additional plans by collaborators on that, and I don't know all of the details, but I think there's plans to do some similar types of data sharing as was done with the farm GTEx.
So just to make sure, Ensemble means then the data coordination center, because Ensemble is not not one-to-one.
Yeah,
it's the FAANG Data Coordination Center because of the metadata.
Yeah, the plan is to have it go through the data coordination center. Yeah, thanks for that clarification.
Good luck with submission.
Yes, thank you.
Krista?
And I need to thank Zhiliang Hu because he's done a lot of work to help with that at Iowa State.
Okay, I have a question regarding methods. So you had just mentioned that you are planning to make the data available. Are you also planning to make the methods openly available for the community?
Yeah, that's a great question. So in terms of the methods, I think there's plans to get those available at present. I don't know the full details, but as much as possible, we're going to make those data available.
Very good. Well, your homework tonight, as well as having a great meal, I think is to think about tomorrow afternoon there's a panel discussion, and I think we've heard some very great talks today which have emphasized the role of community in sustainability. Sustainability is something that is a big challenge for us in animal agriculture, and so again I'd like to have you thinking about the discussion tomorrow and coming forward with your ideas in terms of how we can build on these fantastic projects. Thank you very much, James. Thank you all, everybody, for today's session. I think it's been remarkable.